Hi everyone, this is Nicole Dyer, and this is the Research Like a Pro with DNA question and answer series. And in today's segment, Diana discusses DNA segment tools fundamentals and goes through some examples of working with chromosome browsers and triangulation and figuring out if a segment is maternal or paternal when shared with a specific match, and just some basics about that, and also some warnings about using small segments. So this is an excerpt from a recent Research Like a Pro with DNA office hours session. So hopefully this is helpful for you to get started with DNA segment tools.
So we're going to talk today about exploring segment tools and methodology. And this is an important one because we do need to know how to use segment tools. You may not need to use those in every single project, but it's good to know when you might want to use them and how to use them. So what is the reason you would work with DNA segments? We don't have those available at Ancestry, and so you may never have worked with those at the other websites, and you may have thought it's not worth it to even worry about. But when you come to a sticky problem, it could be that segments really have the answer. So here are three things that I thought of, which would be a good reason to work with some segments. It can add further proof to a hypothesis through segment triangulation. So you may have discovered a nice cluster of DNA matches. They all share with one another, and the documents all work with the DNA, and you would like to just have that final little bit of proof. So you might want to find a few triangulations on DNA segments that could add that little bit extra proof. You might also just want to discover the ancestor who provided a specific segment. And if you can do this, then often that can help you identify an unknown DNA match because you see that they are also sharing segments. Maybe not on that specific ancestor, but for sure on a specific line. So sometimes we just know that it's on a certain line and we're not always sure which ancestor that segment comes from. But as we start eliminating other lines, we sometimes can really get quite far back with segment analysis. So it's worthwhile to get started with it, to try it out. And one of the easiest ways you can do that is just going to the company tools and using what they have. So I thought today I would highlight what we've got going on over at Family Tree DNA and their tools. So Family Tree DNA has the Chromosome Browser Compare tool. It's very, very easy to use, but there are a few things that you need to understand before you go to use it. So one of the things that you want to notice right away is that you can change how many centimorgans you want the tool to report for you. So you have this default of 6 plus centimorgans, and usually we want to try to get larger segments. So you really wouldn't want it to report all the little tiny segments you might share with people. So you can either leave it at 6 or even go up a little bit higher if you want to only show segments that you share with people at, say, 10 centimorgans. You can also download the segment data from here so that you could use that in your report or to put in your Airtable base or to use for chromosome mapping at DNA Painter. So you can do a lot of things just on this page. And I'll show you how you get to this page in just a minute. And then you also have some sections that are grayed out or have these little cross hatches. And this just means that on this particular part of the chromosome, they don't really have enough information or they don't test for matching on this part. And so you're not going to find any matches when you see a gray area. So how do you get to this neat page? Well, you start off with selecting the chromosome browser, and we are comparing whoever you have selected. So we're comparing myself here, and you can start by selecting up to 7 DNA matches. And so you can compare just between 1 or 2 people. You can get 7 people in there. And you can go right here where it says all matches and change that to paternal or maternal so that you can have that narrowed down a little bit if you have done the bucketing on Family Tree DNA so it knows which ones are maternal and paternal. So on Family Tree DNA, unlike Ancestry, it doesn't automatically know that. You have to go into a first cousin and say, this is maternal. And go into maybe a first cousin once removed, say this is paternal, and the more close matches that you can assign, then it will work its algorithm and assign more. So that can help you to narrow people down. So I wanted to show something interesting here so that you really understand how this works. So I have two first cousins here, John, who's on my maternal line, and Susan, who is on my paternal line. I know them very well. And so you can see we have a lot of shared segments because we are first cousins. We share 937 or 834 centimorgans and longest block. These are some really long segments that we share. And so that's something just for so you're aware, we're going to share a lot. All right, so when I do the analysis, you can see that here's that maternal cousin in blue. All these different blocks here are the maternal cousin, and in red are the paternal cousin. Now, if you didn't know that they were maternal and paternal, two different lines, you might take a look at this right here and say, oh, look, they triangulate with Diana. They're all on this chromosome 3 right here, and they overlap. So there would be three people all sharing DNA on this same portion of the chromosome. But remember, there are two copies for every chromosome, a maternal and a paternal. And the algorithms in the websites do not know how to differentiate between that. So if I didn't know that these were two different lines, I would make an incorrect assumption here that we had triangulation. Now, if I knew absolutely that they were both maternal or both paternal, then I could say, yes, that is a triangulated segment because it would be myself and then maternal and paternal, or I'd have two paternal cousins. So you can see there are three different places where we have that overlap. But in this case, it does not show triangulation. It just merely shows that one is sharing on one of my maternal chromosome, the other sharing on my paternal chromosome. So that can be kind of tricky to understand. And so that just lets us know we've got to do our homework and really understand where these people are coming from. We're trying to do triangulation with them. So um, this little segment right here, this I believe just shows a, a break. It's just however they're showing the visualization of the chromosome. I don't think there's anything really specific. I could be wrong, but I actually don't know why they put the breaks there and what that means. So if anybody knows, they can put it in the chat. Now, one of the other things you can do from this page is go to the detailed segment data, and that will tell you the start and stop points and give you all the very specific pieces of information on the chromosome where you are matching. And you can use this in a table or a visualization, a proof argument as you're trying to put this into your research report, those specifics. So those are sometimes useful, and you can also put this into your Airtable base when you have that. So two different ways you can look at your segment data. Okay, and here's a comment in the chat. I think the small point is called the centromere. Oh, that's probably right. I have heard of that term, so thank you. Okay, so some observations just to really reinforce this, that the chromosome browser cannot distinguish between maternal and paternal copies. It requires additional analysis through known relatives or parental testing for definitive assignment. We have to be careful. And then the other point is that we don't want to over rely on small segments. We know the danger of small segments that they may not be real DNA segments. They could be false positives. And so we want to make sure we're really focusing on something that's a little bit smaller, at least 7 centimorgans. I like to go 10 or 15 even. So, you get to decide for yourself how small you want to go, but you want to be careful with that. So that is our little segment on chromosome data and learning to use that. And this whole idea of the maternal and paternal chromosome copies holds true for, say, MyHeritage as well. And it was for 23andMe when we could get that data for them. So just know that you've got to be careful about that.